Hey everyone, it's Jay. Today I'm going to be talking about something called feedback inhibition, which is a concept we're going to have to understand before we talk about insulin resistance and type 2 diabetes mellitus. To get a feel for what this is, I'm a human and I have to eat to stay alive. So I get a lot of my food at the grocery store. When I go to the grocery store, I don't keep buying food till I have way too much. Instead, I'll check my pantry. If I have enough, like say I have enough cans of beans, then I'll stop buying them. Cells are like humans in that they need ATP to stay alive, but they're not going to make too much. There's a process called feedback inhibition that turns off the production of ATP as ATP levels get too high. Give an example. One of the sources of ATP synthesis in the human body comes from breakdown of glucose. And the first step in the breakdown of glucose for ATP is called glycolysis. When ATP levels drop, glycolysis is turned on. There's an enzyme cascade that results in the synthesis of ATP and some other things we're going to ignore. Now, the enzyme that catalyzes one of the first steps in glycolysis is abbreviated PFK. As ATP levels rise, ATP can bind to the enzyme PFK and turn it off. This means that as ATP levels accumulate, the rate of glycolysis drops and ATP synthesis drops as well. Feedback inhibition refers to this idea that the product of a process turns off its own synthesis, and we'll see examples of feedback inhibition all over cells. Another example of feedback inhibition we see with insulin signaling. When insulin binds its receptor, there are signals sent through the cytoplasm that result in glucose transporters moving to the plasma membrane of a cell, and then glucose travels from the interstitial space into the cell. This is going to lower blood glucose. Other signals from the insulin receptor tell the cell to store energy. So there will be fat synthesis and protein synthesis. Now, the products of those processes, increased fat storage, are going to turn off the synthesis of fat through feedback inhibition. But if we start to turn off signals from the insulin receptor, we're also going to turn off signals that send glucose transporters to the plasma membrane of this cell. This means when the insulin receptor is turned on, fewer glucose transporters are inserted into the plasma membrane of a cell, less glucose travels into the cell, more stays in the blood, and this predisposes an individual to diabetes mellitus. The reduced effect of insulin signaling from the receptor is called insulin resistance. Feedback inhibition is why obesity is a risk factor for insulin resistance and type 2 diabetes mellitus. Okay, quick summary. Feedback inhibition refers to the process by which the product of an enzyme cascade turns off that enzyme cascade. This helps limit the overproduction of certain molecules. As we move forward talking about diabetes mellitus, we have to understand how feedback inhibition plays a role in turning off signals from the insulin receptor to understand insulin resistance and the development of type 2 diabetes mellitus. Up next, we're going to dive into diabetes mellitus, two different types, and we'll distinguish where insulin resistance plays a role as opposed to dysfunction in pancreatic beta cells.